I'm one of the vertebrate paleontologists on campus, and I hunt polar dinosaurs. A lot of my research focuses on trying to understand how dinosaurs were distributed throughout the world, and believe it or not, we now know that there were dinosaurs present in the polar regions, both the Arctic and the Antarctic. So I spend a portion of my summer going to the Arctic Circle to find out what dinosaurs were there, whether they were juveniles or just adults, and whether or not they might have migrated back and forth from this region. Because just like today in, say, Svalbard, Norway, we see 3 months of complete blackness where there's no sun and no primary plant productivity. So we really don't understand how these animals were able to survive in such an extreme environment. And so the goal of this research is to try to get a better understanding of the life history on Earth in extreme places, so we can try to understand how animals were able to inhabit extreme environments, the kind of thing that has relevance today as we look at major changes in climate throughout the world. That's how I spend my summers. But the rest of the year I work on a number of other projects, particularly related to my major research interest about the vertebrate head. So this part of our body has got a lot of stuff going on. We see with it, we taste, we eat, we hear, we breathe, and we think with our heads. And so there's a lot of conflicting developmental and evolutionary patterns at play in the way that our heads have come to be formed, particularly within vertebrates, but also within mammals and within humans. And so part of my research uses a new technique that allows us to see soft tissues in micro CT scans so that we can look at how certain structures like the brain have changed across big changes in evolutionary time. This gives us not only the ability to see within the brain without dissecting or damaging it, but it allows us to then reconstruct structures like the kinds of structures that our students in gross anatomy, first-year medical students, will be looking at in their cadaver dissections. We can digitally render these and create 3D atlases, and it allows our students a better ability to understand the tissues that they see in their first patient during their first year.